This is the industry update panel. We have 4 guests here with us today, and so I would like to introduce them. I'll introduce them one at a time, and they can give you a little bit of background on them, and we can move forward. This session is being recorded, so you can watch it at a later time as well. So I think I'll just go the way they're on my panel here. So my first person on my panel here is Asif Packer. So Asif is with Swan Bio. So Asif, I'll let you talk to the group.
Thank you. And Bob, is this the introduction or you want me to go through my presentation too?
Just introduction at this time.
Okay. Hello, everyone. I'm Asif Packer. I'm a neurologist and neuromuscular physician. I'm working at Swan Bio, where we are developing AAV gene therapy for a leukodystrophy. It's adrenomyeloneuropathy. That's an adult phenotype of adrenoleukodystrophy. Prior to Swan Bio, I was early in my career was part of Bluebird Bio, where I designed this clinical trial, the gene therapy study to treat childhood cerebral form of ALD. Prior to joining Bluebird Bio, I was at Kennedy Krieger Institute, where working with Dr. Hugo Moser and part of the team that ran the Lorenzo Zoil clinical trial in adult patients with adrenomyeloneuropathy. And thank you, Bob, for inviting me, and it's a pleasure to speak with you and with all patients and families today.
All right, very good. Thanks, Asif. All right, the next person on my list is Ann Smith from Ionis. So Ann, I'll let you introduce yourself and the company.
Thanks, Bob. I appreciate it. So I'm Ann Smith. I'm in clinical development at Ionis Pharmaceuticals. I've been here about 7 years, but spent about 20 of my career in this kind of work. So we develop antisense oligonucleotides, or ASOs. We have maybe 40 drugs at some stage in development. We have a large pipeline in neuro, and it's very exciting. We've just started a clinical trial in Alexander disease.
Great, that's exciting. I know the families are excited to have that going on. So that's good. I'm sure you had a good meeting with the group earlier. So the next panelist is Shika Denyo. She's with Trevir Therapeutics, and I will let her tell you a little bit about herself and the Trevir Corporation.
Hello, everyone. My name is Shika Dunyo, and I'm Associate Director of Patient Advocacy at Trevir Therapeutics. If you're not familiar with Trevir, we're a small biotech company based in San Diego, where we are developing therapies for people with rare diseases. We work in the rare liver, rare kidney, and rare metabolic disease states, including CTX and Zellweger. And as we like to say at Trevir, we're in rare for life. So we are fully committed to making sure that we partner and develop therapies for people with rare diseases.
All right, very good. Well, the first thing I want to do is thank you all for coming. Asif, I'll kind of start with you. I know that you've got a little bit of a presentation that you'd like to share. Why don't you go ahead and do that, and then we'll move into the questions that we have to go through today.
Definitely. And okay, so I thought I can go through my program, our gene therapy program, and that is that is focused on ALD, but the significance or the relevance of this program to other leukodystrophies is there, and we thought that it also, if when appropriate, can apply to other leukodystrophies too. So let me start here. As I was saying earlier, I worked at Swan Bio. Swan Bio is a small biotech company based in Philadelphia, and we develop AAV9 gene therapy for AMN. And I want to give you a short introduction to gene therapy, and this slide summarizes most of it. Genes are made up of DNA, which are blueprints to build proteins that make our body work. For example, in ALD, ABCD1 protein is built by the gene affected in ALD, and this protein helps break down very long chain fatty acids. Gene therapy introduces a working version of the faulty ABCD1 gene into the cells, and this is done using a vector, which is often derived from a virus, but the viral genes have been removed. Only therapeutic genes are delivered into the cells. The modified cells then produce the protein that was missing or defective prior to the treatment. This vector can be delivered either outside of the body, ex vivo. So if you look at this slide, on the right side here is ex vivo gene therapy, or it can be directly injected into the body, what's called in vivo gene therapy. Ex vivo gene therapy, you may have heard Bluebird Bio's ongoing clinical trial or therapy. Bluebird Bio uses ex vivo gene therapy technique and uses lentiviral mediated gene transduction to treat childhood cerebral form of ALD. So they take, and I'll go into more details there. This approach is not appropriate for AMN. So for AMN, a direct infusion of gene therapy using, this time we're using AAV vector to deliver that gene is used for AMN. The ex vivo approach, so let me see, hold on, my computer is still frozen. Okay. The AAV9, let me focus on AMN today. Okay, AAV9, because this is more relevant to rest of the leukodystrophies too. The ex vivo technique is purely for cerebral form. The AAV9 vector carry the working copy of ABCD1 gene, is injected directly in the CSF, the fluid that surrounds the spinal cord and brain. Now, I'm talking about ABCD1, that's for ALD. For another leukodystrophy where it makes sense, it could be another gene that could be delivered using AAV, either it's 9 or 2 or 5, some other vector. For ALD, with this injection, AAV9 vector will deliver the functional copy of ABCD1 to the cells of spinal cord, where most of the pathology in AMN is focused, localized. This will provide cells the opportunity to produce healthy ABCD1 protein to slow or stop the disease. Let me go to the next slide. Gene therapy, especially the AAV or AAV9 gene therapy, is not a new modality. AAV9 gene therapy is approved therapy for patients with spinal muscular atrophy. And Anne can tell us a lot. Anne's group is working on Spinraza, but this is Zolgensma. I was thinking of Spinraza there. AAV9 gene therapy is approved therapy for spinal muscular atrophy, similar to AMN. SMA is a disease of neurons. In these patients, motor neurons are affected, and AAV9 gene therapy can slow down or stop the disease. So if you see here, the motor neurons are located in the corticospinal tracts here, and in AMN, this is both the corticospinal tract, the motor tracts, and position and proprioception sensation in dorsal column is affected. So we think that this therapy, as it works with SMA, can work for AMN too. But this is a comparison. Now we know also a team in Boston tested the AAV9 gene therapy in the mouse model of ALD. After the treatment, the healthy ABCD1 gene was observed in the cells across the whole spinal cord of the mice. And this also led to reduction of elevated very long chain fatty acids. So if you look at this graph here, y-axis is a ratio of very long chain fatty acids. Higher it is, the elevated it is, and we want to bring them down. And you can see that by intravenous injection, the very long chain fatty acids have decreased. In ALD, very specific to ALD, their very long chain fatty acids are up, and that's the reason that drives the pathology in this and manifestations in this disease. So the aim is to bring those down. What are we doing? So we have Swan Bio is a small company. We are in preclinical stage. So we do not have we do not have a lot of clinical programs. We are working a lot with animal studies at this point. We have treatment studies where we will have treatment studies where we will investigate the AAV9 gene therapy. Currently, we're completing animal studies, the safety studies needed to start the first in human study, and I will reach out to you once we are ready to enroll patients in this study. The second study that we're working on is a natural history study, which helps us understand the progression of AMN disease. This will also help physicians and regulators to decide about efficacy and safety of gene therapy. This study has been started earlier this year. In Europe, the study is ongoing in Amsterdam, and if you're interested, you should contact Amsterdam Medical Center and team there. Germany will soon start enrollment. In the US, this study will be conducted in Stanford, Utah, Boston, Baltimore, and Florida. And these clinics will be ready to enroll in the fall of this year. So these are the treatment studies or natural history studies. In parallel, we're working with another company called Autobahn. So in collaboration with Autobahn, we are developing a patient-reported outcome measure. And this effort is going on at University of Rochester. So why it is important? The AMN specific endpoint first of all, it will be AMN specific endpoint and outcome measure. It can be used in the future clinical trials of AMN, irrespective of the product. So it could be used in gene therapy, it could be used by smaller molecules, by Autobahn or Viking or other companies which are bringing their compounds to clinical trial. Because it is a patient-reported outcome measures, based on our experience with these outcome measures, we know that we will need a smaller number of patients, a fewer number of patients to see an effect of the treatment, or if there's no effect, no effect. So with a smaller number of patients, we can do trials, and that will be very helpful. Right now, they have completed their phase 1, but keep an eye out for calls from University of Rochester. They will reach out to you through ALD Connect and through Bob and his team at United Leukodystrophy Foundation, because we need more than 400 patients for our phase 2 to complete this project. So we need your help for development of patient-reported outcome measures. Just keep an eye out. Thank you, Bob. And I can take questions. I mean, after the presentations are done, or as you see appropriate.
All right, very good. Thank you very much. And can you kind of expand, when you had your opening comments, you were talking about ASO. Can you kind of expand on what that exactly is for the lay people who are watching this?
Sure, happy to. And this is a great time to talk about it, because there can be a lot of confusion about what's an ASO versus what's gene therapy. So this is a perfect opportunity to explain kind of what's similar and what's different. They are both modalities that intend to interfere when you have a protein problem. So not enough of a particular protein, too much of a particular protein. As Asif just mentioned, a gene therapy is one approach to try to replace the ability for your body to make that protein. And ASOs are another modality that can be used in that way. They're both beautifully specific. They're sort of the product of our advances in science in the genomic era, and they can both be designed and developed in a way that really specifically addresses the core genetic issue in a disease, which is beautiful for us to see as scientists. And it's made possible by this understanding of the central dogma of molecular biology, where the DNA, as Asif was describing, contains all the genes to make all the proteins your body needs. But there's an intermediary in that step where a transient, a temporary product is made called RNA. So DNA actually is the instructions to make RNA, which are the instructions to make proteins. And gene therapy is either introducing a new bit of DNA, or it's attempting to make changes to existing DNA. Where ASOs are different is that we're acting on the RNA. So we're acting on that intermediate message. We don't touch the DNA. It stays safe and protected in your cells. And if we need to make more of a particular protein, we will develop an ASO that works in a way that it upregulates the amount of RNA, so more protein is made. If we want to suppress the amount of protein that's made, like in Alexander disease, we're going to introduce an ASO that chews up RNA so that less protein is made. And one of the advantages here is that it's titratable. It's easily titratable. You give a lot more ASO, you'll get a lot more of the change that you're going for. One of the disadvantages is that ASOs are treatments, not cures. So this would not be a one-time shot. Gene therapy is envisioned as a permanent change. ASOs are not. You would need to take an ASO in the same way you take a drug chronically for any other chronic condition. So there are pluses and minuses to each approach, and it's really been very interesting to watch them come along. ASOs had a bit of a head start, maybe a decade ahead of gene therapies. So Zolgensma, which is the gene therapy for SMA, came a few years after Spinraza, which was an ASO for SMA, and they're both effective drugs. And as drugs go, they have pluses and minuses, but we're able to kind of attack the same targets, again, because of this new knowledge of the genetic causes of disease and, gene modifiers that affect disease progression. This information is just coming in like waves, and it's a wonderful place for us to be playing now in science, because it gives us a lot more targets and ways to design better drugs and have better trials that will ultimately lead to better drugs. Is that helpful, Bob?
That's perfect.
Good.
That's very good. I think that'll be some good information for people in the future. I kind of want to switch gears here a little bit. We've got some things we want to get kind of some company feelings out and some thoughts. So I think I'll start with Shika, and I guess first question would be and I'll have all of the panelists respond to these questions. So, but what do you see as advances that maybe will help benefit not only the leukodystrophy community, but maybe also the company's point of view on these advances as well?
Sure. Yeah. Given the company that I have on this call, it sounds like we're all, working hard to come up with therapies that are going to change the lives of people with leukodystrophies. And gene therapy obviously is kind of the newest innovation that's going to be bringing promise to so many rare disease communities. When I'm talking, or when I'm thinking about the perspective of Trevir, we have a therapy in clinical trials for CTX, cerebrotendinous xanthomatosis, and we're hopeful that will be another advancement that will bring promise to so many families. So if those who are excuse me, from that community who are online and are interested in learning more about the trial, they can visit clinicaltrials.gov.
Okay, good. Appreciate giving the clinical trials a little shout out. Lasif, would you like to respond to that question?
Bob, I was reading the ULF, another section. What was the question? Can you say it again?
Okay. What do you see as advances that will help benefit the leukodystrophy community, and especially from your company's point of view?
Over this time, we know over 2 decades of my career, the leukodystrophy field has covered a long distance. As we learn more about the genes itself, how they function, and how that genes express proteins that come to function, to come to the right side of the body, right in the cell, and do what they're supposed to do. These understandings is opening a box of different new treatments, whether these are gene therapy in the form of gene therapies where we add gene or add gene, or through the antisense oligonucleotides or other smaller molecules that can work upstream and downstream in those mechanisms to help these patients. So I think the genetic, the learning more about genes, learning more about how they work and how each mutation in these phenotypes or in these diseases are broken, and where the which mechanism is not working fine, helps us understand the mechanism and help us find a solution that is fit for each leukodystrophy. Thank you.
Yeah, that's good. Yeah, I've been around a long time to watch the changes happen over the years, so I
think,
know exactly
yeah,
what's going on.
I agree. I think that it was in earlier 2000 where we just knew about the mutations, but we didn't know how they work and how can we target them. And now, over last 10 years, not only we know how to target that gene in the sense that if you want to add a healthy gene, and how can we add that, and which cells and which region in brain are the true target for that replacement, because as we know, not whole brain or whole spinal cord may not be the target tissue for one leukodystrophy. So depending on each leukodystrophy, a different approach is taken. And that understanding has, it is a lot of new research in that understanding, and that has opened up new fields for this treatment, for these patients.
Good. Thanks, Asif. And so what do you see as advances that will benefit the leukodystrophy community, and especially from your company's point of view?
Yeah, well, I think we're singing from the same hymnal here. It's the advances in genomics that have enabled Ionis to have success in ASOs and have brought gene therapies that will are ushering in this era of precision medicine, understanding at its core what causes disease and what genetic variants modify time of onset, age of onset, disease progression. These understandings are just coming fast and furious and opening up all new ways to approach targeted treatments.
That's, as the old saying goes, it's a very exciting time. Got a quick question here if you want to answer. And Shika's maybe at somewhat of a disadvantage because she is my patient advocacy person, and I do have two science backed people. So she is a disadvantage. So she is there here to answer as many questions, but I thought this one just kind of fit in here really well. Each of you are obviously working in different leukodystrophies with different approaches. In a best case scenario, the treatments work. Do you see these treatments being applied to other leukodystrophies?
I can start. I can tell you about gene therapy. So for gene therapy, the answer is no, because these are customized and customized therapies, and they have a one specific gene that's usually missing in one leukodystrophy. For each leukodystrophy, we will have to have a different gene. But when we talk about smaller molecules, when you listen to other presentations about working on oxidative stress or working on some other pathways that may be common between leukodystrophies. If that's the case, a treatment may work on more than one leukodystrophies. But it's very much dependent on the mechanism of that drug and also the pathophysiology of that disease. So, I mean, it's very hard to answer this question in general, in a very general. There's no general answer to this question.
Yeah, and I'm going to agree. I think we can conceive of ASOs that could be useful across different populations, but that's not the low-hanging fruit. The easy targets, and no target is easy, but the easy ones are these monogenic diseases where you can trace all symptoms, all manifestations back to a problem with one gene, with one protein. And in that case, the beauty of it is the specificity. But as we think broader about other targets, if you want to hit a DNA repair enzyme, if you want to hit other proteins that are involved in the process of disease, it's conceivable that they could be useful in different diseases. But again, those aren't the easy ones. Those aren't the really good shots on goal that we all have right now.
Very good. Thank you. Appreciate that. Shika, this next question is kind of a two-pronged question. You know, what can foundations like the ULF do to help advance drug therapies? And then the second part would be, what about families? How can they help in this whole process?
Sure. I would say that foundations like ULF and excuse me, other patient advocacy groups should continue to do what they're doing, which is to support and educate their communities on the disease itself, advocacy, how to partner with your physicians to improve health outcomes. I'd also say it's important to educate your community on the drug development process. I think a lot of people just don't really understand what the development of a drug looks like, and the stage of research and all of the stages of research, the clinical trial process in general. I think I think a lot of people still don't have a good grasp on it. So I think where nonprofits can help is to provide that education. And I would say that as long as people feel educated and empowered, they can help us do a better job in what we're doing and be empowered, thoughtful partners to us as we try and develop therapies and gather insights to better understand the patient experience.
Yeah, and I would agree with that. We really value these lovely relationships with foundations like ULF. They're so important to advancing therapies, from getting new drugs into clinical testing all the way to ensuring that regulators like the FDA and insurance companies understand your needs as patients. So we love that you hold meetings like this one. We can't develop therapies in a vacuum. We need to hear directly from the patient community about the diagnostic journey, about aspects of disease that bother you. We do this work to improve patients' lives, so we need to hear what patients want out of these drugs. Maybe second, that the ULF encourages patient communities to band together is awesome. Many voices are stronger than one. And the more you understand, as was just mentioned, the drug development process, you can look for opportunities for the patient voice to get in there, get in front of FDA and tell them about some of these rare diseases that they will never have heard of. So they're ready when drugs come along to understand the impact that those drugs could have. And another advantage here is that we count on ULF to help us communicate with families about leukodystrophy, about the drugs that we're developing. So where we have that partnership, where as a company we have information we want to share and we can blast out through you, that's really helpful.
And I agree with both Sika and I'm so another part that they have taken the best parts of the discussion. I can add is, one part that we really struggle in when we're developing therapies is reaching out to patients and identifying patients. So foundations can help us doing that. And with that, I mean is that if we can work on, a foundation can work and develop a database of the patient population in US, if they have an approach to Canada or Europe, if they have a list or databases of patients. And I'm asking them, and the reason behind that is twofold. One is when we open up studies, we want to reach out to patients that are in all corners of the US, and we may not have that ability to do it once study is open. So if Bob already keeps it, and he does, a long list of where all AMN patients are, where all ALD patients are. So when study comes up, she says, well, I know we can find you 500 patients, we can find you 100 patients or 20 patients. Doesn't matter. That is a big help to companies and to drug development. And second main thing is, the real is, in smaller companies, when we are looking at different indications and seeing which indication should we target, which disease we should work on, one of the factors to consider is how many patients are there for that indication, and are they getting diagnosed in time? So although if a disease affects a large number of patients, but if we cannot find them on time, or we cannot find them at all, the chances that a company develop drug for that disease are lower compared to a smaller disease, but patients groups are very organized and every patient is counted for, and they can go up and say, well, we have 1,000 patients community, and developing a drug for 1,000 patients is maybe, or 10,000 patients could be very different than developing a drug for 200 patients. But on statistics, the number may seem higher, but in reality, we cannot find it. So I think having a databases of patients and foundations can work with the patients to have those connections, that will be very helpful.
Well, one of the things that we at the ULF are partners with is the Global Leukodystrophy Initiative Clinical Trials Network that has received a grant from the NIH. And so we are one of the approximately 40 foundations that are involved in that whole process. From that process, we've actually developed a patient advocacy group program so that we can continue seeing what collaborations we can do together to work together in more ways than one. And so I think that's one of the things we really want to try and do as a foundation. So that's one of the things we do. And I mean, we're looking for a lot of different ways that we can improve collaboration between all the groups. And I be totally honest, I really love what I'm seeing in the space now, because there's a lot of collaborating happening, and the science researchers realize that they need to collaborate better, and so that's really one of the biggest wins that I'm seeing, especially over the years. So you kind of sit here and wait for a lot of years for things to start aligning a little bit, and they seem to be doing a good job of alignment. So that's really good. So I guess I'll come over to Shika and start with her with this one. So what advice do you have for families that are hoping for your company to have a solution to their disease?
I would say I would encourage families to continue to learn about their disease so that they're empowered. I'd also encourage them to learn about advocacy and relationship building. You just noted that you're seeing so much more collaboration across the space. And I think that patient advocacy groups, in addition to families themselves, should be collaborators in the process. But it takes courage, it takes education and being really knowledgeable about your disease. And so I think that families need to work in empowering themselves by educating themselves, by learning about advocacy, by having grit. So just to give you the folks on the call a little bit more about me, I used to work at NORD. I was the director of education programs there. So I've worked with and have seen and met many, many people from the rare disease community. And I think what I've noticed is that the families that tend to get a lot done are the ones that have a ton of grit and they don't give up. And so I would, that would be the advice that I would give anyone trying to fight for their family.
Great answer. So I guess I'll just kind of go around. I guess, Anne, I'll let you respond from your perspective.
Sure. So I guess I would give two bits of advice. Stay connected and understand that the drug development process is slow. And the latter being, we are working hard to address these issues, and we will stay connected with you and give you updates. But just the process of designing clinical trials, getting approvals to do them, enrolling them, completing them, it's slow, and you have to have stamina. But by the stay connected, I mean connected to the patient community, which can be through excellent organizations like ULF. Also be connected to the physician community. These are important for keeping up to date on progress in drug development, and you're also likely to learn about opportunities to help, whether that's being involved in a clinical trial or participating on an advisory board for a company, or even filling out a survey. We do these steps to bring in data that we need to make good decisions. So there are any number of activities that the patient community can get engaged in, big and small, that can have meaningful impacts on our ability to be efficient in drug development.
And what do you think, kind of a timeline, everybody wants everything done real fast, because you talk about it slow, can you maybe just kind of give some sort of little timeline just so people will have a little bit of extra information on understanding that?
Oh gosh. Well, if I think about our Alexander disease program, which is just one example, and I'd love to hear Asif give another example just to give some context. I think we had our early promising data in animal models about 12 years ago, 10 to 12 years ago, the kind of data in an animal that makes you think, yeah, this could really work. We could really get there with this. And then it's a matter of convincing yourself that those data are real, repeating some experiments with ASOs and also with gene therapy to an extent. The drug discovery process can actually be more efficient than in some other areas because we know exactly what we're trying to build. Whereas if you're looking for, say, a small molecule that's going to impact the amount of protein that's made, that's a much more difficult task. So we could spend a couple of years identifying, designing the drug, and then a couple of years of what are called toxicology tests, which are, every company's got to do these, regulators require them to understand the properties of your drug before you put it in a human. So you can see these couple of years add up, and 10 years might be a nice ambitious goal from, that spark of an idea to maybe being able to get into the clinic. But that's just, gosh, I don't feel like I can speak for the industry. I'd love to hear Asif's answer to that too.
Go for it, Asif.
We'll put you on the
I
spot.
agree with that. Yes, I agree. I fully agree with you, and I think there's no one correct answer to that. So I can tell you about clinic first because that's easiest for me. Once the first patient is treated, so we already in the patient. If we are really on fast track and everything is working just great and drug is doing good, it will take good 7 years to 8 years to get that drug approved. I think just the way of the usually what we know for leukodystrophies, or these are diseases where it takes time to show an effect. If it was diabetes where we can give insulin and measure blood glucose, that time can shrink. But for not slower, but difficult diseases, and with variability that comes with these diseases within patients and between patients, it takes longer. So give at least 7 years once first patient is treated. Before that, it all depends on how much time has gone to discovery. It takes good 1 year. I'm talking about here really short timelines. 1 year to do toxicology studies and animal studies just to fulfill FDA's basic requirement of going to humans. And then, then there's a lot of earlier work. And if it's a small molecule, you have phase 1 studies and phase 2 studies to do those. 10 years is a best case scenario, but it usually takes a lot more. And what Sika said, is the grit, is the patience, is the persistence, is very important. It is. We would like to bring drugs in a more rapid, fast manner, but it's just not possible. It's just not possible. This is not that we do not have, or FDA doesn't. FDA is doing something. It's just the way this things works. It takes time. And you would like to have that time. I understand that if a person who has with a patient, with a family member may not feel that, but you would not like to have drugs approved that are not safe, that can harm you more than what your loved one is going through now. So it's when you look from top, this is a time that we take to make sure that you get the right dose, you get the right route, and you get the good possible effects from a drug that you're hoping for.
I think I kind of remember back, what was that, about 2012 when you started working for Bluebird, and you got thrown into the fire pretty good because I'm not so sure if it might not have been Florian that bailed on that. And he made you give the speech and tried to explain, quote unquote, not explain the gene therapy was taking place. And so we're 11 years out from that meeting, but when that was really being processed. So that was really, that kind of gives people an idea because they're still not
approved
in production. Technically, the product still have compassionate use, but it's still not on the market yet. So
Bob, so it's a good example. So I can give you that example because it will give a good idea. In July of 2013, I think we were near Chicago in DeKalb at that time, or Baltimore, one of those sites.
It was DeKalb.
DeKalb, yes. Where I came in, explained, but that was a time when we treated our very first patient with childhood cerebral this phenotype with gene therapy. With 2021, so it's now 8 years out, and we just have submitted the package to Europe and working on FDA. So good 8 years. And the work that had gone before. So in 2006-07, when Patrick O'Bourke treated 4 patients. So work started way earlier, even though a treatment that shows 90% efficacy. It's such a strong efficacy with a very good safety profile. It is taking more than a decade to get it approved and become available.
Yeah, so that's why we have to be persistent and patient all in one same thing. Got a question from Dean is, how can we apply what's been demonstrated in the COVID research? Been very fast from clinical trials. What can we do in leukodystrophy space? Is it possible? Maybe I should.
The COVID vaccines had the advantage of precedent. So we know how to develop vaccines, right? There was a new technology used, but it's well documented, well understood technology. So while it wasn't an easy undertaking, it's much easier undertaking than attempting to address a genetic disease where there is no precedent. Sometimes there have been no clinical trials in these populations. The endpoints aren't established. As I mentioned before, the regulators don't know what they're looking at when it first comes to them. So there's a groundwork and there's an education that needs to happen along the way. I'm not sure, maybe Asif can think of a way to exploit the process of vaccine development to help us. If you've got it, I'd love to hear it, but I'm not sure there's much learnings we can take from there to help us out.
I think there was a lot of political pressure and what FDA did. And I can tell you that because a lot of our meetings, requests, and our communication with FDA was postponed or delayed because they rerouted their staff to work on COVID projects. So there was a lot of pressure on FDA to assign large teams on COVID protocols and things. So I think some of that worked, and getting approvals or emergency approvals in a week is unheard of. And I mean, unless there's a big political push behind that, that aspect itself is challenging. And drug development part is, I agree with you. This was a unique case that I don't think may set a precedent for us. I'm not sure about that.
So I do want to say, I don't want FDA to come off as bad guys here. I have experience with them being very generous and collaborative and active in spaces where there is an unmet need. So these rare diseases, some of them pediatric, I've seen drugs approved in a 3-month period, 12 weeks from application to approval, which is really fast. So rest do know that the teams there they're humans and they're compassionate and they're professionals. And if the data are there to support the approval, they can act fast for us too.
No, yeah, I fully agree. I think that FDA for rare diseases, for diseases where such a high unmet need is, like the leukodystrophies, they go out of their way to help us. I have on multiple occasions when we're working when I was working with gene therapy in early 2000s, so 2013, 14, when this concept was new and we were working with FDA, they will come back to us with a more to ask us and ask help of explaining to them what it is, what we're seeing, so the clinical relevance of what we were learning in those studies, and how can we put it in context with the existing knowledge and things we have. So I think FDA is very open. They want to help us out. I think in COVID it proved in 1 week. That 1 week is extreme, but 3 months is 4 months is a good time. And to give you an idea, if you go with a smaller molecule, let's say for schizophrenia and depression, they will take 9 months to 12 months. So 3 months is very fast. 3 months for FDA is unheard of. So they are out there to help us. It's just the amount of evidence that we have to collect to convince regulators, physicians, and families. It takes time.
Christy had a question that really fits right into this, asking, isn't the NIH and the FDA looking at platforms to deliver gene therapies faster?
Bob, you asked me a question?
Yeah, well, whoever wants to answer that. Just question about the NIH and FDA looking at platforms to deliver gene therapies faster. Do you know of any or have any information on that?
I didn't understand this question well. Do they have, great.
Christy, you want to ask him the question? I'll just let you come on live and ask him the question.
Yeah, thanks, Bob. So I know that I attended some NIH and FDA meetings where they talked about being able to package up certain diseases together, to potentially deliver on gene therapies faster, making more like a rinse and repeat process of being able to, so that it wouldn't be so long from beginning to end, since many of these things are delivered in a similar fashion through AAV. And whether, we could move faster, if, especially on the smaller genes. So that's,
yes.
Just what I was asking.
Yeah, Christian, yeah, I'm aware of that effort, and a very good question. So I want to say that when you talk to high level of FDA, they would agree and they said that we should do it. But when you go down and work with the teams, it takes a little longer. So that being said, but it's just that they have all the good intentions, but it just takes it takes a little work on both sides to realize it. But let me step back and let me walk you through this process. The way we can do it faster is we take the AAV and we swap genes of interest. So for ALD, we can say put in ABCD1. For MLD, you can say can the ARSA-A gene, or the gene that codes for ARSA protein, we can put it in and then use it. That is easy part, but then the delivery and dose is very difficult. That's where each molecule has to go through its own process. So for ALD, you can give it to CSF through intrathecal delivery. For metachromatic leukodystrophy, probably you need it more in the brain than in spinal cord. So then they have to put a burr hole and want to give it through that part. So very different approaches. Doses depend on dose of a gene therapy is not like Tylenol or not like a drug that you can say you can do it half or full. It has to be tailored for that indication, and that part takes to work. That part needs to work on. So now on in principle, it seems that gene therapy could be simpler and FDA can do something, but the work they ask you to do, and not FDA, the work that's required to do is it takes almost the same time for each disease. It's very difficult to have that approach for AAV. I hope I answered your question.
Yes, thank you. It's not simple.
It's not simple.
Yeah, I guess a follow-up question would be, is there any particular piece of the process that has the ability to become faster if certain technology was available? I know when I attended several sessions, they talked about just the ability to produce the AAV, production-wise was a challenge.
So what FDA has helped us do, and the community has done. So the gene therapy has two parts. One is the vector, the carrier, AAV, whichever serotype it could be, and second is the gene. So what part that has been facilitated is that for AAV, we do not need to do 200 or 400 or several mice studies because we know that part. So that part, sometimes we do not have to repeat. We just can refer to previous work and move to the gene of interest and work on the safety and dose of that part. So those, if you think of 10 years ago, everybody has to do their AAV work, then the gene work, and then they go to humans. Now, some of that work has been reduced. So there are developments. And there are developments around immunosuppression that goes in. So we know rather than reinventing the wheel on immunosuppression for each indication and for each product, now we know for AAV9, what do we know? How can we do it? Depending on, we know 1 time, it could be 2 times. Those things are something that are lessons that we have learned, and we can use it for other diseases, for other leukodystrophies.
Okay. Perfect. Dean had another question to kind of build off of gene therapy. Could gene therapy from a given biopharma, and not for the first therapy, but for subsequent therapies, be a platform building block? So I'm thinking that's probably at you, Asif.
Yes. So I think I tried to answer it in earlier thing is, when I look at my own program in Swan Bio, so we are looking at diseases, neuromuscular indications, looking at neurology. That all helps because in natural history we can say diseases like AMN, which has spasticity and paraplegia. Paraplegia means weakness of the legs and spasticity of the legs. We can combine that part together and can say we can do natural histories that study different indications in one natural history study. So that's one. Second, when we're using the gene therapy, we can use the AAV9. I'm using an example of my own program, AAV9. So I have already figured out AAV9 part of it. Now I have to figure out which gene to give, and then has to say which dose will go in. So that's a kind of we're trying to build that platform in our company so that we can add more genes and quickly test whether that works or not in each indication. And but I was saying is, but it is, the challenges are that our genes are so complex that sometimes in some indication you can give as much gene as you want and it will work. for some other indications, you have to dose them. If you give them a little bit more, it becomes toxic, and it then is harmful. If you give a little bit less, it's not enough to show an efficacy. I think we are working with those challenges of selecting the right dose, and that's that's a big hurdle towards a platform gene therapy, a one program where you can just swap the DNA.
Okay, thanks, Asif. We're getting down close to time, and I've kind of got one more final question for each of the members. So I'll give Asif a little break, and I'll pop over to Shika and just ask, do you have anything else that you'd like to share with our community today?
I see I'm muted. Thank you so much for inviting me, and I tried to hold my own with two scientists on the panel, but we are incredibly proud to be partners to ULF, and I feel really grateful to work closely with you, Bob, and the CTX community. It's been incredibly inspirational to work with such an amazing group. I myself, as I mentioned, at NORD, developed educational programs and resources. I didn't get to work as closely in the community as I do now. And so we are very proud of our long relationship with ULF, and hope that that continues for many years to come. And so I look forward to seeing what's in store for ULF and for the CTX community. You're muted, Bob.
I didn't want anything in my background to pop up. So we appreciate you as well. And Trevir has been a very good partner for us over the years, and we do look forward to keeping relationship rolling. So, Anne, I will ask you to answer the question if there's anything else you want to share with the group.
Sure. And also thank you for the invitation. Shika, it was good to meet you, and you held your own. You did well. Awesome. Good to meet you too. So let me just express my gratitude for the passion and the involvement of the ULF and the patient communities. What you do inspires us. And when you contribute to research, really anything from filling out a survey to participating in a clinical trial, you're making a difference and helping us to be better informed and more effective. So know that your involvement is a contribution to developing therapies. So thank you.
Well, thank you, Anne. And like I say, we've enjoyed working with Ionis over the last several years, and we look forward to continuing the relationships. That's great. So Asif, I'll give you the last word from the speakers.
Thank you, Bob, and thank you, Keely and Sika and the rest of the group. And this was very informative 2 days for me, and giving me this opportunity to share our program. I think we look forward to meeting together in person. Today with gene therapy, I see there's a good interest in gene therapy. Maybe sometime I can come prepared in the sense that I can bring more materials to go through about genetic therapies and have a session for the leukodystrophy group, because we have similar sections for ALD. Now we can expand it to leukodystrophies, and we can discuss this further. I just want to thank you all.
All right. Thanks, Asif. Obviously, you and I have known each other for a long time, so it's we go back to the Kennedy Krieger days, so it's been a while. So, but we appreciate Swan Bio and all that they've done for us as well, and I know that'll continue in the future. So, one thing I want to let people know is that these recordings will be ready probably in mid-July. So just kind of watch our website for information when those come out, and we'll send some e-blasts out as well. So we'll keep you informed on what's going on. So other than that, hopefully we're planning on being in person in Chicago next year, the last week of June. So I believe 23rd to the 25th, I believe are the dates. We look forward to having a great big reunion. So get your quote unquote tickets early because they might get sold out. We just never know. So with that, thank you, panel. I appreciate all that you contributed to this. And for those who were able to be on here live, thank you for the questions that you developed. And so until we meet again, we'll see you later. Take care. Bye-bye.
Thank you.
All right.
Thank you.